with this condition, is there a group that we should be in communication with or something like that? And, for some genes, there are not. And those folks, I have to sort of go one out, asking individual families to get them connected. So this is so meaningful and so important that you're doing this. And so just congratulations. I'm going to run through some slides with you. Just I want to leave some time because my colleague, Dr. Mackinson, I want to be able to talk a little bit in more detail about some of the work that's going on in the lab, but especially with the samples that many of us spent so much time today going through the process of collecting, what their utility is, why they're particularly special, and what we might be able to learn using them. But just quickly, I'm a pediatric epileptologist I have a background training in science and also medicine. And so my goal is really to to see patients and then not stop with whatever we're able to do in the clinic today, but to go back to the laboratory and try to push the envelope and see where we can take things in the future to make lives better. Because we're often sort of stuck with, managing seizures and other symptomatic sort of band-aids for conditions, and there isn't that sort of precision therapeutic that would tackle the actual underlying cause of the disease. So my work largely involves mouse modeling, but this is our group. We're part of the labs that you saw today is the Center for Translational Research in Neurodevelopmental Disease, or the CTR&D. And we are a group of scientists who are committed to pushing the envelope on these different disease genes. And Dr. Mackinson and myself are very invested in CSNK2B. And these are some of the sort of techniques that collectively our different laboratories are capable of doing, everything from EEGs in mice to recordings from mouse pups during the development, to modeling human nerve cells in a dish, and even miniature brains, and recording from them and learning about how genetic changes affect nerve function, protein function, all the way up to network level changes that affect entire organisms in the case of the mouse. And so CSNK2B is like a number of genetic causes of neurodevelopmental disability and epilepsy. We all have differences in our genes that make us different from one another. But some of our genes don't change between people, and even looking across species, like from mouse to people, or even from worms to mice to people, don't change so much. And CSNK2B is that kind of a gene. And when a change is new, it's a de novo change. And those kinds of changes are often the ones that we associate with disease, with genes like CSNK2B. These are some of the, this is what we reported back in 2020. Now there are even more, right? Now there are even more genetic changes that have been reported in the literature. And this is a picture of the gene. This is just showing you the, in a linear way, sort of a stretch of residues that code for different things. But when the protein gets made, when it forms something, not a line, but a complex sort of globular structure, and that's depicted over here with the beta units here. And two beta units come together with two alpha units to make something called a kinase. And kinases are a signaling molecule within cells that run around and tag things. And when you get a tag, you might behave different. So this is a protein that has the potential to change the behavior of whole groups of other proteins, running around tagging them. And it might tag them only under certain circumstances, and it might be different in one type of cell and another. And the tagging part is on the alpha subunit. So the beta subunit has something to do with holding the whole thing together, and maybe to do with making sure it goes to the right places. But that's one of the aspects of the research that we need to learn more about. And so these are the different types of changes. Sorry about the DNA got messed up here. This is DNA, right? So these are, everybody has 2 copies of the CSNK2B gene. And in most people, there's no change. In some people, they have the asterisk, and that means that there's a change that makes it so that you don't make that copy. You don't make anything from that copy of the gene, so you have half. And then there's some people who have a change, but it's a change that makes it makes the product different than what it's expected to be. So that's the example of the one where you just don't make enough, only making it from 1 copy. And then instead of the red triangle depicted here, you make a green circle. And we don't know what that does. Maybe the green circle doesn't work as well. Maybe the green circle actually gums up the works and makes things worse in a certain way. Or maybe the green circle does something completely new that is unexpected. And so all of both of those types of changes have been reported in patients with CSNK2B. And I think it's common. So we were talking about this a little bit earlier with some of you, but I think it's more common than is appreciated. We sequenced a bunch of people here, and all of the genes that, first of all, this is sequencing people with epilepsy, and of 68 people who had a genetic diagnosis, there were 48 genes. So there's a lot of different causes of epilepsy. That's called heterogeneity. There's a lot of that. There weren't so many genes that we got more than 1 hit on that gene, right? But the ones that there were, these are well-known genes. These are well-known genes. SCN1A, the cause of Dravet syndrome, right? KCNQ2 is a cause of KCNQ2 encephalopathy. These are very common. Among the rare, these are very common conditions that are well described. All of these across the board. This is lissencephaly. This is something called Soda syndrome. These are all very well known. CSNK2B is the most important epilepsy gene, I think, that people, doctors don't know about yet. And for whatever reason, it's sort of been late to discover, late to be added to the panels. And so people are only now sort of like coming in droves with this diagnosis. I think you're going to see more. We happen to be sort of on the leading edge of this because of that sequencing study that I was just telling you about. And so we've identified a number of patients here very early on, right as the gene was first being reported. And then we connected with a lot of other families and clinicians across the country and even internationally to put together a series. And what we found is that there's a real range of effects on individuals. And you probably know this. The affected patients, the kids are not all the same. They're very different. There's a big range. So some kids, I will tell you, are not able to walk or talk at one end of the spectrum, right? And they may have bad epilepsy with recurrent seizures that bring them into the hospital, and we can't get them out of the hospital, and then they're discharged and they only come back again in a week or 2. There are other people who don't have epilepsy at all. Maybe 20% of patients aren't having seizures. And then there's a whole spectrum in between. Some kids learn to talk, some kids don't learn to talk. We don't know what determines those things. That's a big mystery, and it's one of the reasons why we need to do the natural history study that we're doing. What we do know is for people who develop the epilepsy, the seizures most often start early in life. So this is not all patients, but this is just of the patients who go on to develop epilepsy. Most of them develop it before 3 years of age. So I know some folks out there have kids with epilepsy that started later. And that's less common, but that's another aspect of the spectrum. It can happen, and some people apparently don't have epilepsy at all. And just finally, to drill home this point, they did a study of people who go into the hospital repeatedly with status epilepticus, and they tried to see if there were any genes responsible for those kids' presentations. Most of the time, it's not a genetic situation, but sometimes it is. And when it was in that model that they put together to look for genes, they found MECP2, which you might know as Rett syndrome, SCN8A, which again is one of those genes that we know in epilepsy is very well known, well described, sodium channel gene. SCN1A, again, Dravet syndrome, and CSNK2B, again, showing up where all of these other very famous players are there. And so again, I think it's a very important thing that people are not aware of. So if I had to sort of summarize the big picture for the disease, early onset epilepsy, often but not always, right? Not always early onset, not always epilepsy. Low tone and motor delays, I think, are very, very common. Language delays and cognitive disability, and some impact on language. That's kind of the overall picture with a lot of variability. And these two different mechanisms for changing our CSNK2B so that people develop these problems. But we don't really know how those changes in those genes result in disease. And we don't know whether or not there's more than one thing going on. So for the missense, where the red triangle becomes a green circle, there's possibly other things happening other than just you don't have enough of the kinase working properly. And then what's the relationship to the alpha subunit? So some of the alpha subunit is encoded by another gene called CSNK2A1. And that is a gene that was worked on here by Wendy Chung, and is responsible for a whole nother set of patients. Maybe it's a related condition, and maybe there's a way to work together. And we've been in communication with that group to try to collaborate. So speaking of collaboration, so I'm going to pivot to what we've been doing and what we want to do. So this is a paper that came out of the Schwartz lab at the University of Connecticut. So I told you that CK2B is CK2 beta is a kinase, right? And the beta subunit is what CSNK2B encodes. So CK2 has a way of recognizing what it's supposed to stick tags on. So when it runs around, it's looking at different proteins and it recognizes different sequences. The Schwartz lab, working on the alpha subunit of CK2, CSNK2A1, they figured out using a lot of biochemistry that I'm not trained in and I don't really understand. They identified the changes to the recognition sequences brought about by the changes by the mutations to the gene. In other words, they figured out how the mutations change the CK2 activity. And you can see if this is the number of proteins that normally get a tag under the normal circumstances in the red circle, some of those, so the blue circle is with the common mutation, some of those still happen, but a lot of them don't. And there's a lot of new things that happen. And so we don't know the relationship yet between what these changes do to the kinase activity and, for instance, what not having enough beta subunit does, right? And so one big collaboration we have, and one way in which the samples collected today are going to be useful, is with the University of Connecticut doing similar studies to look at how the mutations that change the beta subunit CSNK2B, how those change the kinase activity, and how that's the same or different from what's happening with CSNK2A1. I'm just spouting a lot of alphabet, but people following?
Yeah.
Ask questions. So then coming back to what the group here wants to do, right? So we've made a conditional mouse model. So one way, if you're going to test therapeutics that you're going to develop, or you're going to try to understand the changes at the level of an entire brain, one way to do that is to model it in mice. Mice are tricky because it turns out that if you knock out CSNK2B completely from mice, they die before they're born. And if you take out one copy, so sort of like what a lot of patients have, they kind of do okay, actually. And this is a common problem with this type of mutation in mice, they compensate. They say, we're just going to make more of the copy we do have. And so you can't win, right? Either they're dead before they're born, or they're fine, right? So what we've done is to actually generate what's called a conditional mouse model that allows us to turn it off only in certain parts of the body, like the brain, and see what's happening when we do that. And we can do all kinds of different manipulations to study that. So it gives us some traction over that problem. Almost done, I swear. Okay. So then, and Dr. Mackinson is going to talk more about this. But the other thing we can do with those skin cells is we can turn back the clock on them and have them make other types of tissues. So like neurons, and even allow those neurons to sort of normally assemble into little miniature sections of brain in a dish, little chunks of brain. And that allows us to look at how those neurons are working and how they work together or don't work together due to the changes brought about by the gene mutations. Okay, what's the point of all of that, right? Well, the whole point, at least from my perspective, the science is nice, but I became a doctor because I wanted to help people, and that's why I went back to the laboratory too. And so the whole point is to try to move forward with treatments. And what we really need is a way to try to address the underlying problem, right? And so what we're working on in the lab and what we do routinely at this point for conditions is to do just that, to try to devise a strategy to alter the disease course by replacing a gene or taking it away if it's doing something bad, or possibly both. And this is the viral vector that is being used commonly for gene therapies that are being developed now. It seems safe. And CSNK2B fits inside nicely because it's a relatively small gene. And so part of the point of having all these model systems is so that if there's a problem in the nerve cells, we should be able to give the virus and fix it if the virus is working properly to replace the copy of CSNK2B. And this is already, we're well underway. Abigail in the back of the room has already designed our first attempt. And then the final thing to say is that I also talked to you about collecting data. It's not just so that we can learn about the spectrum of disease, because I think it's important that we do have a grip on that. But my hope is that we can identify some outcome measures for use in future trials, because if a therapeutic like this comes out or some other treatment becomes available, it would be nice to be able to look at an outcome measure that doesn't take years, like development of language. Okay, we'll give this now, and in a few years, we'll see if it worked. Right? That's a problem. It's not just a problem for us committing to giving children something that, you know, is experimental for that period of time. It's also a problem for industry because they have to have some turnaround, some idea that their investment is actually worthwhile. And so my hope is that by looking at the EEG patients, that will be able to identify that biomarker for a short-term outcome for some of the future trials that will come. So that's it in a nutshell. The updates are we're actually submitting the CSNK2B gene reviews today, not today, but this week. Maureen, who you met earlier, was around. She's been instrumental in that, along with another genetic counselor, Natalie Lippa. There's the Schwartz Lab collaboration, neurons and brain organoids, mouse models, and gene therapy. And this is all my thank yous, and especially thank you to the foundation and all the patients for being so engaged in this work. And a special thanks to Heather and Frank Jackson for supporting the research that we're getting into now. Is that fast enough?
Yeah, that's great.
Okay. So I'm going to ask Chris to come up and talk because he's much more eloquent about brain organoids and neurons and assembloids and all kinds of other wild science. Yeah.
All right, let's see if I can get this out.
Sorry.
You get a preview. The next slide.
When you click the display stuff, yeah, we have to, yes.
All right, that looks much better. Well, thank you all for coming.
And you?
I'm more of a basic scientist, so I have a PhD, not an MD, but I've been working on different models of epilepsy since I started my PhD well over 10 years, almost 15 years ago now. I've studied a number of the genes that were Tristan's presentation, so SCN1A. Right now, we study SCN8A, and we have a number of collaborations on other genes as well. And I'll give you somewhat of an update on what we're doing on CSNK2B right now, but really I'm going to use kind of bits and pieces from ongoing experiments to show you what our plan is for kind of the future, because this is just getting started. But, you know, one thing I want to make clear is having the samples that you all have provided today is absolutely critical to do this work. And without it, there really is no project. It's the beginning of what we need to study the gene and to try to understand what's going on in the disorder. So thank you. All righty, so this is more or less what I kind of just told you. So early on in my graduate work, mainly what we were doing is identifying putative disease-causing genes and then introducing those genes into the same position in the mouse genome, and then we'd study those mice. So we would do EEGs to look for seizures. We would do various types of neurophysiology to look at how the cells and the circuits within the brain are are acting. We've knocked the gene out of different cell types, which is something I'll talk about a little bit later to see like which is the cell type that's really important. And that's something that's, important for therapies, because you might be able to get away with only delivering, say, a gene therapy to a small part of the brain. That's much easier than targeting everything. But you really need to know kind of what's the connection between the mutation and then something functional, right? Like how does the brain work in the context of the mutation? And to do that, you need a good model, right? So during my PhD, the standard model for mice, and this made a lot of sense for Dravet syndrome, which is caused by SCN1A, and for SCN8A developmental disorders, because basically the gene is the same between the mouse and the human. And so you find a mutation in a clinical population, you can find the same position in the mouse genome and introduce that mutation in the mouse, and then the mice end up having seizures. So it kind of makes sense, right? It's the same mutation, you put it into the mouse, and the mouse has a seizure. That's a good model, right? But that's not always the case, and mice are different than people in many ways. And so, you don't always want to, I think we got rather lucky actually with the Dravet models and the SCN8 models. There are other genes where there either isn't the exact same location between the, mice and humans. So there's really nothing to, there's no one-to-one there. And then there's some cases where you introduce the mutation and you don't see the same things. Right? And so during my my postdoc, I became really interested in developing human-based models. So this was then like a few years after these discoveries that allowed scientists to take essentially any cell from your body, reprogram it to sort of like turn back the clock, developmentally speaking, and turn that cell into what's called an induced stem cell, right? So these are different than like embryonic stem cells. These are cells like the skin cells, and then you turn them into a stem cell, right? And then once you have the stem cell, there's been this just huge field and growing field of researchers who figure out what factors and what environment you need to put that stem cell into in order to get it to become certain types of other cells, right? So there's some researchers take an induced stem cell and make liver, or will make heart, or will make skin, or will make brain. And then even within brain, there's all kinds of different brain cells. So you can further kind of, direct the path of that cell, not just to brain, but to inhibitory cells or excitatory cells or the cerebellum or the cortex and so forth. And that's kind of where the field is right now. Some of these protocols are really well worked out and efficient. Some of them aren't. What we're, luckily for us here, the cortical protocols are some of the best described out there, and that's what we're going to use. And it makes a lot of sense to start there for CSNK2B. But that's sort of a long way of saying that this was one of the first papers describing a cortical organoid that I published a while ago. And then this is a really neat one. So we found that like once you can take the stem cells and make two different brain regions, you can stick them together, and then they form connections between those regions. And so this is a lot like how connections form between different brain regions during development, right? You've got the left side of your brain and the right side of the brain are connected through the corpus callosum, right? You can make two cortical organoids and stick them together, and you get a connection between them. That's a way to like model that part of the circuit. So anyway, that was the first demonstration that you could do multi-region work with organoids. And then the last benefit of them really is that you can keep them indefinitely. So cells in a dish are often sort of unhealthy, right? They don't really want to be in a dish. They haven't evolved to be in a dish, right? They're made to be in a brain, and that's where they're happiest. And while the organoid isn't keeping those cells in a floating ball isn't perfect, it at least allows you to keep them in a healthy state for many, many months. And so in this paper, what we did was simply ask the question, what happens to, molecularly, right, to the cells in the organoid if you just track them for, like a period of time that would match like human gestation, so many months, right? We took them out well over a year. And what we found is that the trajectory of the gene expression changes in the organoid actually matches pretty well what you'd expect from human development over that time period. So they are changing slowly over time. It's not like you make it and it's a fixed thing. It actually continues to develop in some ways that reflect.
Can you like reuse it? Like, can you grow it over and over again? Like
Um,
from the same, because it's a stem cell, can you do that?
Yeah, exactly. So you take the stem cells.
Yeah.
And you do plate those, right? You make sure they're healthy, but you can expand them endlessly,
Okay.
really.
Okay,
And then we freeze them down in little groups, right? And
Yeah.
then we just pull them out of the freezer as needed.
Yeah.
Expand them. Basically, we collect them into these little wells. And then from that, an organoid pops up from each well. So it's easy to expand, right?
Yeah.
If you want 1,000 of them,
Yeah.
just put them in an agro well with 1,000. If you want 5, then put them in a well with 5. It really, it's a powerful approach in some ways, because once you have the stem cells and they're good stem cells, it's easy to make a lot of these organoids for all kinds of different studies. The other disadvantage in some ways is because they're so like human-like in their development, it takes a really long time. Right, so a mouse, mouse lives a few years, and its early development is, weeks, right?
Yeah.
These things, to get real like brain activity in the organoid, it takes months. Usually we start measuring that at around day 90 in vitro. So those are some of the things that we're working on. And this is the general overview of the approach that the lab takes. So we have a pointer for you.
Nobody sees a pointer. Are you there?
Laser point my virtual.
Hi, Chuck.
So this is you guys right here. And these are the cells, and we're going to turn them into stem cells. And then we're going to, we may or may not do this part. I think we will want to correct some of them. So there's kind of two different ways to, major ways to like compare, right? Because what we want to do is figure out what's similar between all of the patient samples and, how do they differ from the controls. But these cells are sort of like people. We're all different, even though, they're from your children, they're not going to be exactly the same. So one thing we're going to try to do is take some of the samples that have the mutation and correct it, right, just in the cells, so that you have the cell line with the mutation and then the exact same cell line, but with that mutation corrected. And that's in addition to the other controls, the parental controls, which are also really useful. So that's kind of just like getting the stem cells that we need for the study. We then pattern them into these brain organoids, which vaguely look like this. I'll show you some pictures of these later. And then we do various types of functional work. So we can look at the pattern of gene expression. This is like a plot kind of representing that. We do a lot of functional imaging. So we can look at fluorescent molecules that we express in the organoids that tell you when a given cell is active. And then this is sort of the gold standard for reading out activity in a cell. We actually take I don't know if you saw this during a lab tour in my lab, but we take a glass pipette and find an individual cell and break into the cell to form an electrical connection with it. And then we can measure what that cell is doing. We can electrically You can see if another cell is signaling to it. And it's a really great approach. The only downside is it's one by one, right? So it's a slow approach, whereas imaging and some of these others we get quickly. But by combining these different approaches, some high throughput, some low throughput, you know, it gives us, I think, a better chance of figuring out what's really the most important thing that's going on. And then, you know, we're trying to understand what does the gene do, just as a basic question in neurobiology. And then we want to leverage our insights to develop the treatment. Yeah.
So because all of us have different alterations of the gene, are you going to specifically look at certain alterations and see if certain alterations in fact like some of those things
Yeah.
themselves, like
Yeah, it's a really good question. And I mean, I think what I would like to do in talking with Tristan on that is sort of find variants that tend to cluster in terms of like the phenotypes that you see in the, you know, and then from that, we can see if any of these sort of measures, right, fall into the like track with those groups.
so that like when down the road when somebody tests for it and say, oh, you have a missense of this, and those are either the typical cells or presentation of what happens, is that like a goal or a sight in mind?
Yeah, and also, you know, the hope is that these measurements are informative to what's going on in the disease, right? And that we can use what differences we see here to develop treatments, right? Because we don't have, you know, in an organoid, we don't have behaviors. They don't have seizures. They don't have any, you know, that people and animals have. So we have to use some sort of proxy for that, right? And so it could be a pattern of gene expression, or it could be, you know, too much calcium activity, or too much, you know, or the temporal relationships between, you know, how different cells are active. And then we would put, you know, drugs or try gene therapy. And what we're looking for is to see that phenotype look more like the control groups, right? The edited or the parental groups. And that's sort of a starting place for therapy. So this is just a general sort of question that we always have, right? You know, how do we take something out in the real world and study it in the lab in a way that can help us, you know, figure out what's going on and develop a treatment. And so, what am I doing on time here? I'll try to speed it up. So historically, there were lots of different ways to model epilepsy. You could give the animals, like a normal animal, drugs or, you know, injuries and things like that. The field has mostly, except for studying that type of epilepsy, the field has mostly moved on to using these genetic models, which is like what I described earlier, where you try to find a gene and a variant where the animal has the same variant as the human. So you just knock in the variant and then you can measure the seizures from there. And so this is a real pedigree from a family with GEFS plus, which is on the Dravet syndrome spectrum. And this is something I did during my PhD. We took one of these mutations and put it into the mouse, and that's a seizure that the mouse had from after we knocked in the mutation. And then finally, this is the human approach. So, you know, we take the cells, make an organoid, and as you can see, we can do most of the same types of things, but not everything. You know, we can't do behavior, we can't do EEG. So there's still a role between the, you know, for animal testing. And that's really why we've decided to go down this two-pronged approach, right? We have the animal model and we're going to make the human cells. So this is a little bit technical, I guess, but this is just an example of how we make the conditional knockout animal that Tristan was telling you about. So basically what we've done is we've put these little extra things called LOXP sites around an important part of the gene. And then we cross that animal for these other animals that have this gene called CRE. And that gene is one that we've selected because it has interesting properties. Either it turns on in a cell that we want or at a time point in which we want. But that gene, CRE, basically it's an enzyme that causes the deletion of everything in between those two sites. So by crossing these animals together, you get all these pups of different combinations, right, of these different genes, because it's random how they inherit them. And we take all the pups that have the CRE gene and the LOXP site, they'll be deleted. And all the animals that don't have CRE, they have normal expression of, in this case, CSNK2B. So that's basically how the animal that we made works. And if you really want the details, that's the actual strategy. We knocked out a bunch of different exons, so it's yeah, so
Can we go back to that slide one?
yeah. Okay, yeah. So, right, so I think the first thing we're gonna do here with these animals is instrument them for EEG and see if they have seizures. It's something that Tristan's lab and my lab do, routinely with these other animal models, and it's something that just, I think, needs to be done really with any new model. We also have a really excellent behavior core, and so we have some plans to do to see how the animal behaves. And there's various tasks that can be done from learning and memory to anxiety and so forth. And I think Tristan will be the one to decide which tasks make the most sense in terms of what is, clinically, right? We want this to be something relevant that we test for. And then this is sort of getting back to some of what my lab does. So really, we want to understand how, what's the connection between the mutation in the gene and a neural circuit problem. And so basically, there are all of these sort of complicated connections between different subtypes of cells. That's like an example of a circuit where and we'll use these different tree lines. This is example data from another project to find each of those cells and record from them. And then we can track, how many firing or signaling events they have in response to stimulation, in response to the current that you put into the cell. And that's a good way to figure out. In this case, okay, the green cells are not so affected. Those lines are basically the same, but the purple cells and the red cells are.
Is this always in the gray matter of the brain or?
Yeah.
Okay.
Yeah, that's where we would do these. You can record from any neuron in any part of the brain, but this would be in the gray matter, which would be the cortical layer there. So I want to quickly get to the brain organoid part because I have some cool videos and not that much time. This is a picture of a brain organoid that has green cells are one type of cell and a purple is a different type of cell. So basically, these are stem cell derived. That's the term that they use in the field, microphysiological system. But basically, it means it's active, right? There's physiology there. There's things that are going on. It's not just like static. You can derive them from embryonic or induced pluripotent stem cells. We exclusively use induced pluripotent stem cells. So they're self-assembled, which is very cool. So you get all of this structure in the organoid. Like, you get a little like ventricle in the middle with the progenitor region around that and then neurons around that. So you get layers just naturally. right? It's like the cells as they're developing are communicating with each other and arranging themselves relative to one another, because that's what happens during development. And there are sort of processes that mediate that, that are, that are quite complex. They develop many different types of cells, which is important. And something that I found is that they develop these functional capabilities. So we could do electrophysiology measurements. We can look at cell mobility and neurite outgrowth, which is something that'll be really important for this project. And it's a bit of a noisy slide here, but this is like how we make them. So we make the stem cells and then we plate them in these non-adherent plates, and they lift off and then slowly get bigger until we either slice them or dissociate them or do something to them that we want for the study. And down here is a picture of them at different time points in culture. So you kind of see these little balls of neural tissue getting larger over time. And this is just some very early reviews, some of the first recordings that were done in brain organoid. I did these during my postdoc. And basically it just shows that they have these spikes here are action potentials, which are like the electrical signaling. And then these little blips here are called the synaptic responses. So that's other cells signaling to the cell I'm recording from. And this is the cell trying to signal out to other cells. And if I can play this, I think I have to get out of the laser pointer view. So this is, these are cells migrating through the organoid. You can kind of see each of those as a neuron. And this is just showing that we can capture that aspect of neurodevelopment and see if it's similar or different in the bigger. This is the same type of thing, but zoomed way in. It's showing the cell moving through the organoid. And this is one of our brain organoid cells. And this is a real cell from human fetal tissue. And then lastly, one of the things that we are doing a lot of in the lab now is measuring calcium. And so this is just spontaneous activity. You can see a little blip here and then like a larger events that light up the whole thing. So this whole thing here is one big brain organoid, and it has an indicator in it that causes it to light up whenever calcium comes into the cell. So it's like an indirect way of measuring how active the organoid is. So you might expect, you know, hyperactive organoid to have more of these events than one that is hypoactive. And this is just an analysis of those events. And then this is just another cool one. So we stimulate the organoid. You can see that's an electrode right here. And right when it stems, you see this wave that propagates through the tissue. And then you see a bunch of activity that follows it. And these are all things that we can analyze. We can analyze the properties of that wave, and we can index each of the neurons within the culture. And bring this and look at when one neuron is active, what's the probability that another neuron in this field will be active? And so the idea is if they're communicating with each other, there should be some relationship there.
So you're tracking like the single neurons in the organoid?
Yeah, so
Wow.
each of these like little dots here
Yeah.
is a single neuron. With the other one, it was like the whole organoid view.
yeah.
It's such a low magnification image that you don't really see individual cells. this is zoomed in on just one little section of it. So that you can see yeah, so each of these little kind of sparkles,
Yeah.
see them going, like
Yeah.
each of those is an individual neuron that's active. All right, and so what we really want to do, and this is something that Tristan mentioned, is come up with therapeutic options, right? So everything that I kind of showed before was making the model, using it to read out different things. We are going to define what's similar and what's different, and then we want to use that to benchmark how well different gene therapy strategies are working. So this is one that's a little different than the gene replacement strategy that Tristan mentioned, which makes a ton of sense. But this is another strategy that we're using in my lab, which is basically just to correct the mutation itself. So instead of like adding in a new gene, right, we want to go in and take the gene that's already there and correct it. Right, it's sort of a complementary approach. So we've been working on this for some time. And at least in some cell models, we have a pretty high editing efficiency. So this is approaching, 75 or so percent editing efficiency, which is really high for this kind of thing. And we are just now piloting in organoids expression of the factor. And so each of these blue dots is a different cell, and all the green sort of around it is the all-in-one construct that would be doing the editing. So we're getting really nice coverage there. And that's now a system that we can use to try corrective genome engineering for CSNK2B. We haven't gotten to the point of trying it yet. You can just prove of concept.
Oh, okay.
And so, yeah, I think that's pretty much everything. I just want to highlight again that, the team that we have here is really great for this kind of a project. We have a number of different researchers who are all really focused on different types of developmental epilepsies. And, I think it's a major strength of the project. So this is the strategy that, the center and my lab really takes, which is to merge these different kind of basic and translational, aims into these sort of more cohesive strategy for understanding the disease and trying to do something there. And these are the members of my lab and funded, which is so important. So if y'all have any questions about the strategy or anything else, I'd be happy to take them or write down my email, and I'm, anytime you need it, we can please reach out.
I'm wondering, since you just started with the mice and they're not exhibiting the same symptoms as our kids, and you said sometimes the mouse model doesn't work, so we don't know if this mouse model will work at this.
I don't know.
Yeah.
That's what we gotta do. That's the
Okay.
first thing, the first thing, I think, is to look at the behavior and look at the EEG.
Yeah.
And just match that to what we see. So, and then there are things that we can do there are things that we can do to see if we just have to adjust, like the level of expression, for example, a little bit to get it. So there's a problem when you're dealing with animal models. The mutation they don't live long enough.
Yeah.
And if it's not severe enough.
You don't see the symptoms.
Like where they're all kind of have different thresholds for different species, right? So that might not match up perfectly, even though the model is essentially good. If you could either make it a little more severe, or you could solve the lethality issue. So that's why the free approach, the conditional knockout approach, may end up being really helpful, because we can let the animal develop normally until they're, a week old, and then turn on the deletion and see what happens. So that the rapidly get worse, or they don't okay because there was some developmental thing that didn't happen, and now we can study the later phases of the disease. So these are kind of things we need to see, we need to work out. But it's also why we're not putting all of our eggs in one basket, you know?
Did Dr. Sands say that the mice would like compensate for the like, so they do that naturally? Like
Yeah,
if you're,
that's right. That's the idea, is that they have other CSNK2 genes, right? And one of the other ones will compensate for partial
okay,
loss.
but whereas humans, we don't have that.
Yeah.
Okay. Okay. I was like, oh, why don't we just use what they have and put it in?
Nice.
Like, oh, how they figured out compensating for this already. I'm like, what?
The mice have already figured it out.
Yeah.
Actually, just do what they do.
Yeah, right. Easy. Yeah.
Actually an opportunity.
Yeah.
It's like, oh, they figured it out. Right?
Probably across a lot of different backgrounds.
Oh, okay.
It's frustrating research,
Mhm.
like Wayne Franklin was like, been doing this for decades. For epilepsy genes, a lot of times the most successful models are the ones that change one amino acid rather than the ones that destroy one of the copies.
Okay.
Probably because of the same.
Oh, okay.
You know,
That makes sense.
why I'd like to try both, like the editing approach, and the overexpression approach, well, Tristan was saying, because, you know, if it's just that you need more of the protein, overexpression would work great.
Right, yeah.
But if it's something like a point mutation causes
Right.
that to like get in the way, like
Yeah.
it's binding where it's supposed to and blocking other things from getting there, or
And-
it's pumping up or whatever, you really
Yeah.
want that to be correct.
Corrected.
And so if you correct
That
that
makes sense.
mutation, maybe that'll be more effective in that context.
And like if we were doing the overcompensating route, would you have to do that like every, time frame? Like, is there like, inject something every periodically, or is it like a one and done kind of thing?
That's, whether, there are some studies in the retina that have done overexpression,
Mhm.
and I think they did need to. Let me check on that actually, because I think they're actually okay for years.
Okay.
For like
Yeah.
retinal overexpression of like chromosomes or something.
Okay.
Yeah, it's a good question.
Okay.
So for knockdown approaches like the ASOs that you may have seen, some of the results come out for that. Those do need to be repeated and frequently.
Right, yeah.
Because you're just injecting these oligos into the CSF, it gets knocked down.
Fine, yeah.
Quickly, so it's only there for a short period of time. This is a different strategy, because you're putting genomic information into the cells.
Right.
And so the question is, how long will you get effective kind of production off of that extra gene that you put in before it gets either shut down? And sometimes they essentially don't get shut down, and sometimes they do over time.
And for the gene editing, it would just be like, you just change it and that's it, right? It's just.
The gene editing is permanent.
Yeah.
That's right.
Okay.
Which is.
Are there typically like major side effects for that kind of stuff? I mean, I don't know how many gene editing that have been done successfully.
It's a rapidly evolving field.
Yeah.
I think delivery is the main hurdle right now,
Okay.
right? I mean, there are viral vectors, but if your immune system recognizes the viral vector, then that's a problem. So there's a lot of work in like nanoparticles and some of these other strategies that Tristan was mentioning. But I think the way I'm thinking about it is there's the strategy that we will try to define the outlines of what can work using the models, and then there's implementing.
Yeah.
And there are, teams of people working on gene delivery, because that, if you solve gene delivery, you have something.
So many,
all
yeah.
of these different
Yeah.
groups of people and stuff will use. So there's just a huge amount of effort on gene delivery. So I'm hoping that will be solved. And happy to license their tech, right? To apply the
Right.
strategy using
Yeah.
that delivery. That's kind of how I'm thinking about it in the future.
Oh, right, yeah, okay. Let me stop recording. Okay. Yeah, thank you, guys.
Should we all stand there? Yeah. I think so.